Hello friends, today we'll talk about electron transport chain. Electron transport chain is also known as respiratory chain, and 70% of oxygen consumed by cell is utilized by electron transport chain to produce ATP. So electron transport is a series of reaction which undergoes transport of electron from electron donor to electron acceptor, and this will drive the ATP synthesis. To understand how this mechanism works, we have to first understand what leads to transfer of electron from one place to another, which will result in ATP synthesis. In biological oxidation, there is oxidation of foodstuff, where carbohydrate, proteins and fats, which are complex macromolecules, gets converted into simple micromolecules, that is monosaccharides, amino acids and fatty acids, and they will undergo metabolism to give acetyl coenzyme A. So acetyl coenzyme A can be formed from glucose by undergoing glycolysis. Similarly, acetyl coenzyme A can be produced from protein by deamination and fats by beta oxidation. So the resultant product acetyl coenzyme A is a 2-carbon compound, and this acetyl coenzyme A will enter into the Krebs cycle, where acetyl coenzyme A will be completely oxidized to give 2 molecules of carbon dioxide during the cycle. And during the Krebs cycle, there is production of reduced coenzymes like NADH plus H plus and FADH2. These are reduced coenzymes. So these are high energy molecules, and they are the electron carriers, and they will enter into electron transport chain and will result in ATP synthesis. We will see that how one molecule of NADH will produce 2.5 ATP and one molecule of FADH2 will produce 1.5 ATP. Now we will try to understand the concept of oxidation and reduction. Remember, whenever there is loss of electron, it is said to be oxidation, and whenever there is gain of electron, it is reduction. So for example, the ferrous form of iron, when there is loss of electron, gets converted into ferric form, which is the oxidized form. And when ferric iron gains electron, it gets reduced to ferrous form of iron. So when there is loss of electron, it is oxidation, and when there is gain of electron, it causes reduction. Now we'll talk about redox pair. So what are redox pair? Redox pair is substances which exists in both oxidized and reduced state. These are known as redox pairs. For example, NAD, NADH, FMN, FMNH2, FAD, FADH2. So these are redox pair, where they exist in both oxidized and reduced state. Coming to redox potential or electron transfer potential. So what is redox potential? Redox potential is the tendency of redox pair to lose or gain electron, that is measured quantitatively. So what are the examples of redox potentials? So examples are NAD, NADH, they are at lower redox potential and their value is minus 0.32. FMN, FNH2, it has minus 0.30. Redox potential of FAD, FADH2 is minus 0.22. Coenzyme Q is plus 0.10. Cytochrome C is plus 0.25. Cytochrome A and cytochrome A3 is plus 0.29, and half molecule of oxygen and water is plus 0.82. So NAD, NADH, FMN, FMNH2, FAD, FADH2, they have more tendency to lose electron, and coenzyme Q, cytochrome C, cytochrome A, cytochrome A3, and oxygen molecule and water, they have more tendency to accept electron. Here in electron transport chain, they are arranged in ascending order of redox potential. So electron transfers from negative redox potential, which has greater tendency to lose electron, to positive redox potential, which has greater tendency to accept electron. Now coming to electron transport chain. So electron transport chain is a series of reaction which undergoes transport of electron from electron donor to electron acceptor, that will drive ATP synthesis. So electron transport chain takes place in mitochondria. Which part of mitochondria? Inner mitochondrial membrane and intermembranous space. So if this is a mitochondria, the outer layer is the outer mitochondrial membrane. The inner one, the inner folds, are inner mitochondrial membrane, and the space between the two, the outer mitochondrial membrane and inner mitochondrial membrane, is intermembranous space. Some cellular process which occurs in mitochondria are oxidative phosphorylation, electron transport chain, apoptosis, which is mediated by cytochrome C, production of reactive oxygen species, that is production of ROS. It is also associated with calcium homeostasis, and it is also associated with immunity. As we had already discussed earlier, glycolysis takes place in the cytosol, in the cytoplasm, and in glycolysis, one molecule of glucose will form two molecules of pyruvic acid, and this pyruvic acid will enter the mitochondria to form acetyl coenzyme A after undergoing link reaction. And after acetylation of pyruvic acid, it enters into mitochondrial matrix, where acetyl coenzyme A will enter into citric acid cycle or Krebs cycle, and during this process acetyl coenzyme A will be completely oxidized to give two molecules of carbon dioxide, and there will be release of reduced coenzymes like NADH plus H plus and FADH2. So these are reduced coenzymes, and they are high energy molecules, and they are electron carriers, and these electrons will be transferred from one place to another in electron transport chain. Now if we focus on this part of mitochondria, it will look somewhat like this. So this is the outer mitochondrial membrane. This space is known as the intermembrane space, and this is the inner mitochondrial membrane, and below inner mitochondrial membrane is the mitochondrial matrix. And there are 5 enzyme complexes that are embedded in the inner mitochondrial membrane. So this is the inner mitochondrial membrane, and these are the complex. So there are 5 complexes: complex 1, complex 2, complex 3, complex 4, and complex 5. So there are 5 enzyme complexes that are embedded in the inner mitochondrial membrane. This complex 1 is also known as NADH dehydrogenase ubiquinone oxidoreductase. Ubiquinone is also known as coenzyme Q. So complex 1 is also known as NADH dehydrogenase ubiquinone oxidoreductase. Complex 2 is known as succinate dehydrogenase ubiquinone oxidoreductase, or it is also known as succinate dehydrogenase coenzyme Q oxidoreductase. And complex 3 is known as ubiquinone cytochrome C oxidoreductase, also known as coenzyme Q cytochrome C oxidoreductase. And complex 4 is known as cytochrome C oxidoreductase. And complex 5 is ATP synthase. So complex 1, complex 2, complex 3 and complex 4, they belong to electron transport chain, whereas ATP synthase, that is complex 5, it belongs to oxidative phosphorylation. And we should also note that electron carriers may or may not be a part of inner mitochondrial membrane complexes. So these electron carriers are FMN, that is flavin mononucleotide, COQ, that is coenzyme Q, which is also known as ubiquinone, and there is FES, that is iron sulfur protein, which is associated with FMN and cytochrome B, and there are cytochromes. These cytochromes are copper containing heme protein. So cytochrome have heme protein and copper, so they are also known as copper containing heme protein. So cytochrome is a copper containing heme protein, and cytochrome can be cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. And cytochrome A and cytochrome A3 in combination, they are called as cytochrome oxidase. So cytochrome A and cytochrome A3, they are together known as cytochrome oxidase. And cytochrome C, only cytochrome C is water soluble and easily diffusible, whereas cytochrome B, cytochrome C1, cytochrome A, and cytochrome A3, they are lipid solubles. So they are fixed to inner mitochondrial membrane. So they are part of complexes, but not cytochrome C. So coenzyme Q and cytochrome C, they are not a part of any complexes. So they are not fixed to inner mitochondrial membrane. So they act as electron acceptor or electron carrier. Now coming back to complexes in electron transport chain, we can clearly say that complex 1 has negative redox potential, and compared to complex 1, complex 4 has more positive redox potential. So electrons are traveling along complex 1 to complex 3 to complex 4. So redox potential is important measure factor for transportation of electron. Therefore, electron flows in only one direction from complex 1 to complex 4 because of redox couple with low redox potential is considered to be better electron donor, and those with higher electron potential is better with electron acceptor. So they are electron donor and these are electron acceptor. So complex 1 is also known as NADH dehydrogenase ubiquinone oxidoreductase. So complex 1 has FMN and iron sulfur protein. And now we'll talk about the sequence of transfer of electron in complex 1. So NADH plus H plus will transfer its electron to FMN and will get converted into NAD plus. Now FMN will accept the electron and gets converted into FMNH2. Now FMN will donate the electron to iron sulfur protein, and now this electron will be donated to coenzyme Q, which is a mobile electron carrier. So we can get this NADH from pyruvate dehydrogenase complex. We can also get it from TCA cycle, beta oxidation, and malate-aspartate shuttle. So this is the sequence of electron transfer in complex 1. So during the transfer of electron from complex 1, near about 10 to 12 kilocalories of energy is released, and this energy is utilized to pump 4 protons across the inner mitochondrial membrane into the intermembranous space. So in intermembranous space we have 4 protons right now. Now coming to complex 2. Complex 2 is also known as succinate dehydrogenase ubiquinone oxidoreductase, and this complex has FAD. This complex has FAD and iron sulfur protein. So the sequence of transfer of electron is from succinate. Here succinate will transfer the electron to FAD and will get converted into FADH2. Now they will transfer the electron to iron sulfur protein, and electron will finally be donated to coenzyme Q. So complex 1 and complex 2 both are giving their electron to coenzyme Q. Coenzyme Q is also known as ubiquinone, and it is a mobile electron carrier. So during the transfer of electron from succinate to FAD to iron sulfur to coenzyme Q, there is no pumping of proton, that is, it does not act as a proton pump, because the energy released is not sufficient to drive the proton across the inner mitochondrial membrane. So there is no pumping of proton by complex 2. Now this coenzyme Q will transfer electron to complex 3. This coenzyme Q will transfer the electron taken from complex 1 and complex 2 and will give it to cytochrome B. Now the electron from cytochrome B will be donated to iron sulfur protein, and then to cytochrome C1, and finally to cytochrome C. Here cytochrome C is also a mobile electron carrier. So coenzyme Q will transfer the electron to cytochrome B, then to iron sulfur protein, then to cytochrome C1, and then to cytochrome C. And this cytochrome C will now transfer the electron to complex 4. So during the transfer of electron from complex 3, near about 10 kilocalories of energy is released, and this energy is used to drive 4 protons across the inner mitochondrial membrane into the intermembranous space. So we got 4 protons from complex 1 and 4 protons from complex 3, and complex 2 will not pump any proton into the intermembranous space. And this complex 4 is also known as cytochrome C oxidase. So complex 4 is also known as cytochrome C oxidase. Now the cytochrome C will give the electron to cytochrome A, then A will give its electron to cytochrome A3, and now the de-energized electron will be taken by half molecule of oxygen and will be converted into water molecules. From complex 4, there are pumping of 2 protons outside the inner mitochondrial membrane into the intermembranous space. So as the electrons gets transferred from complex 1 to complex 3 to complex 4, and from complex 2 to complex 3 to complex 4, the electrons will get de-energized. That is, electron will lose its power. In order for electron transport chain to continue functioning, the de-energized electron must be removed. So oxygen acts as a final electron acceptor, removing the de-energized electron to prevent the chain from becoming blocked. So oxygen also binds with free proton in the matrix to form water molecule, and this will remove matrix proton to maintain the hydrogen gradient. So during the transfer of electron from NADH to coenzyme Q to final acceptor from complex 1, complex 3 and complex 4, so total 10 protons are pumped out across the inner mitochondrial membrane into intermembranous space. But during the transfer of electron from FAD or from succinate, first step is skipped. That is why only 6 protons are pumped out during the transfer of electron from complex 2. From NADH we get 10 protons, and from FAD we get only 6 protons. So as there is pumping of electron outside the inner membrane into the intermembranous space, and this inner membrane is impermeable to proton, so there is development of proton gradient outside the membrane. So there is development of proton gradient outside the membrane. So there is more positive outside and more negative inside. So this development of proton gradient is responsible for ATP synthesis. ATP synthase is that complex which will move proton from intermembranous space into mitochondrial matrix. ATP synthase has two subunits, F0 and F1. So ATP synthase is also known as F0 F1 synthase. So when H plus comes to the mitochondrial matrix through complex 5, ADP undergoes phosphorylation in presence of ATP synthase and becomes ATP. This is how ATP is generated. So during the transfer of electron from NADH to coenzyme Q to final acceptor, there are total 10 protons that are pumped out into the intermembranous space. So there are 10 protons from one molecule of NADH, and for every 4 protons 1 ATP is formed. For every 4 proton 1 ATP is formed. So from 1 NADH we will get 2.5 ATP. And similarly, during the transfer of electron from FAD or from succinate, first step is skipped. That is why only 6 protons are pumped outside the inner mitochondrial membrane into the intermembranous space. So we get 6 proton which is pumped from one molecule of FADH2. So for every 4 protons 1 ATP is produced. Therefore from 1 FADH2 we get 1.5 ATP. Now we will talk about how much ATP is produced during the metabolism of glucose. So one movement of glucose through glycolysis down to electron transport chain can generate total of 30 to 32 ATP. So from glycolysis we get 2 direct ATP and 2 molecules of NADH, and this NADH will go into electron transport chain, and 1 molecule of NADH will give 2.5 ATP. So 2 molecule of NADH will give total 5 ATP. In glycolysis 1 molecule of glucose will give 2 molecules of pyruvic acid. So 1 molecule of pyruvic acid will form 1 NADH and 2 molecules of pyruvic acid will form 2 NADH. So we get 2 into 2.5 ATP to get total 5 ATP. Now this pyruvate will enter into TCA cycle. Now TCA cycle, it takes 2 turns. So this TCA cycle will spin twice. So for a single spin of TCA cycle it will produce 1 ATP. In 2 turns it will produce 2 ATP. So this is a direct ATP produced in Krebs cycle. Now one turn of Krebs cycle will produce 3 NADH. So 2 turns of Krebs cycle will produce 6 NADH. So 6 into 2.5 will give you 15 ATP. And in one turn of Krebs cycle there is 1 molecule of FADH2 formed. So in 2 turn of TCA cycle 2 molecules of FADH2 is formed. So 2 FADH2 will form 3 ATP. So one movement through glycolysis down to electron transport chain can generate 30 to 32 ATP. 30 to 32 ATPs. So in conclusion we can say hydrogen carriers donate high energy electron to electron transport chain located on inner mitochondrial membrane. As the electron moves through the chain, they lose energy which is transferred to the electron carriers within the chain. The electron carriers use this energy to pump proton from the matrix into the intermembranous space. The accumulation of proton in intermembranous space creates an electrochemical gradient or proton motive force. So protons returns to the matrix by transmembrane enzyme ATP synthase. This diffusion of ions is called chemiosmosis. All the ions passes through ATP synthase and they trigger a phosphorylation which produces ATP from ADP and inorganic phosphate. Now the de-energized electron are removed from the chain by oxygen. allowing new high energy electron to enter the chain. And oxygen also binds matrix proton to form water. This will maintain the hydrogen gradient by removing proton from the matrix. So this was all about electron transport chain. Now we will talk about inhibitors of electron transport chain in our next video. Thank you and keep watching our videos. And if you like the video, please do subscribe our channel and share it with your friends. Thank you.